So, welcome back. Today I'm just going to review the mechanisms that eukaryotic organisms utilize in order to regulate their gene expression. So these are topics that we have already covered in class, but I just wanted to put this out there so that you would have them if you wanted to look back on them. Or, if anybody happened to miss class that day, they have this accessible to them. So the regulation mechanisms occur at each step of our protein synthesis central dogma here. So whether they be the molecular steps or the processing steps, there are ways in which our cells can start or stop the process of gene expression at any point. Right? And so our cells don't always want to transcribe and translate every gene that we have available. We don't always need all the proteins that we can make. Right? So most of our cells have a lot of the genes already shut off. For instance, our eyeball cells do not need to be making the proteins that are responsible for the functions of the heart. Right? So even though our eyeball cells have all of the DNA that we possess in our entire genome, you only want the genes that are related to the function of that cell to be turned on. And these regulation mechanisms are how we go about doing that. On top of that, there are certain conditions that must be met for most proteins to need to be expressed. Right? And so we can regulate whether or not our proteins are being made dependent upon the circumstances that our cells find themselves in. So that is what we're going to explore in this video, just looking at some of those mechanisms. Right. So we are starting at the level of DNA. And so we are talking physically about the structure of the DNA itself and some modifications that we can make to that structure to regulate whether or not any specific gene is going to be expressed. And there are two main ways in which our cells regulate gene expression at this level. The first of which is methylation of the DNA itself. We take a molecule called a methyl, we stick it right on the DNA strand, and it blocks transcription. So this methylation process, literally it just is stuck there and it keeps the RNA polymerase from coming in and engaging with the gene. RNA polymerase can't engage with the gene, transcription never happens. Right? Transcription never happens, you never get this protein made. So this methylation process is really effective at blocking the expression of any given gene that is methylated. And one of the most important aspects of this mechanism is that the methylation patterns are maintained during mitosis. So anytime a parent cell has a specific methylation pattern with specific genes shut off, every daughter cell that is made in that cell line will preserve that methylation pattern. So this is what allows our cells to copy themselves as the same type of cell. So when an eyeball cell does mitosis, it makes two eyeball cells. Those two eyeball cells are only going to be expressing eyeball genes. So this is very important in the process of cell differentiation. When we get a zygote, which is just a single baseless cell, it is what it is, and it divides up into a bunch of different cells, that differentiation process where those cells gain their unique qualities is due in large part to this methylation of specific genes. Right? So that's a very permanent change. Through science, we can remove methylation, but it doesn't naturally occur very often within our body. Alternatively, we can also methylate or acetylate the tails of the histone proteins that we find in the chromosome structure. So if you recall the chromosome structure that we talked about in the couple videos ago, right? There are these histone proteins that work kind of like fishing reels for the chromosome. Right? And so the chromosome will wrap around the histone a couple times and then come out the other side. And so there's a bunch of these histone proteins along the length of the chromosome. And now if we tighten the DNA strand around this histone, those genes that are inside that tight ball are inaccessible to the RNA polymerase. Right? And so if we tighten this coil, those genes will never be expressed. Okay? Similarly, if we loosen that coil around that histone, genes that might have been inaccessible beforehand are now accessible to the RNA polymerase. And so by adding either a methyl or an acetyl to these tails of these histones, we can change the way in which the chromosome is coiled around that histone. Adding a methyl to the tail will tighten that coiling and it will reduce the accessibility of those genes. Whereas adding acetyl to the histone tail will loosen the coiling and that promotes transcription. These changes are temporary. Right? So this depends on the conditions of the cells. If there are conditions that want to stop the expression of specific genes, right, the methyls will come in and get on those histone tails. If there are conditions that inspire the cell to promote the transcription of a specific gene, the acetyls will come along and acetylate those tails and loosen it up, and that transcription can begin. So moving onward to a transcription level regulation for eukaryotic organisms, right, which is different than the prokaryotic mechanism, which was the operon that we've covered. Okay. So this is specifically going to be eukaryotic organism regulation. We have to take a peek at the anatomy of the gene, which we covered in another video in the past, right? So we have the promoter region directly upstream from the actual structural gene. So these lines represent the coding of the gene that is actually going to make the protein that we're looking at. Right? So immediately in front of that, there's a region that has a specific sequence that is conducive to the RNA polymerase attaching to this region. Right? So the RNA polymerase can't just attach willy-nilly wherever it wants. It needs to attach to a specific area, and in this case, that area is called the promoter. So the promoter is the area that the RNA polymerase can actually attach to. Part of the promoter is the proximal control elements. Okay? Way upstream, and we also have the distal control elements. So distal control elements, proximal control elements. And then in between the two, there's a long strand of non-coding DNA. Okay? Just inert, chilling out DNA, not coding for anything. So there's a few steps in getting this gene expressed. Okay, so we're starting as a straight line, can't express it yet. Okay. By the end of this story, we're going to have the RNA polymerase attached to this promoter and rocking and rolling and transcribing this gene. So the first thing that needs to happen is cellular conditions have to be favorable for this gene to be expressed, and those favorable conditions will result in the generation of an activator transcription factor. Okay. So transcription factors are a whole group of molecules that engage with the control elements that lead up to the process of transcription. There are positive transcription factors, which will speed up or turn on the transcription, and there are negative transcription factors, which will slow down or turn off the transcription of this gene. Okay. Most of the ones that I'm doing in this model are positive transcription factors. So this activated transcription factor is a positive transcription factor. It is going to engage with the distal control element. Okay. Once that activator is engaged with the distal control element, it will trigger another protein to come in, okay, and engage with the non-coding region that lies between the control elements. This is a DNA bending protein. It's named that because it bends DNA and it's a protein. Amazing. Okay. Once that DNA bending protein is engaged with this non-coding region, it will bend the DNA up. Okay. So it's bent right here, you see that. The importance of this bending is that it now puts the distal control element in line with the proximal control element. So they're in line with one another. Once they are nice and lined up, you get this giant complex of proteins that builds basically a bridge between the two control elements. They are a mediary protein complex and it's built from a bunch of smaller proteins. And that protein complex has a very high affinity for other transcription factors. And so the transcription factors will come floating in, and this protein complex will grab them, and it will introduce those transcription factors to the proximal control element. Once those positive transcription factors are engaged with the proximal control element, it will change the shape of the promoter so that the promoter now fully fits with the RNA polymerase. The RNA polymerase can attach on and go down and transcribe this gene. And so that is not possible until these positive transcription factors have engaged with the proximal control element. Cool. Yeah. This is also where negative transcription factors can come into play, because they can block the positive transcription factors from engaging with the proximal control element, or they can just slow them down. And so we can regulate how fast this is going, or we can turn it off if something supersedes the signal to turn it on. And so if cell condition A says, hey, make protein B, but cell condition 2 says, hey, wait a second, we really don't want protein B right now, cell condition 2 can send a signal to stop this process midway. Okay? So it's never outside of control. All the way through, we can always stop the process of gene expression. So now we have our RNA. Our RNA has been made already. We finished transcription and we are in possession of an RNA. So the RNA that's made immediately after transcription is finished is a primary transcript. It has a copy of every nitrogenous base in RNA form that is complementary to every single nitrogenous base that was represented in the gene of the DNA. That primary transcript is not our final mRNA. It needs some modifications to occur prior to it being the actual finished job. And the most important modification for this storyline is the intron and exon splicing. Okay? And so not every base pair is going to be used in the final mRNA. Some of them are going to be cut out. And so we have regions that are going to stay and are going to be expressed, and we call those exons. And we have regions that are going to be cut out, and we call those introns. Now, the exons and the introns are not finite things, right? They can be changed. They can be altered. There's nothing physical about them that makes them an exon or an intron. They're only considered introns and exons when they are cut out or when they stay in. And the molecule that's making the decisions about what's getting cut out and what's staying in is the spliceosome. And so this is a large protein complex with some RNAs involved as well that go through and sort out what part of this primary transcript is going to stay as an exon and what parts are going to be cut out as an intron. And depending on cellular conditions, there's different molecules attached to the spliceosome, and those different molecules change the shape slightly, and those changes in shape are what are identifying what parts are exons and introns. So this will be based on cellular conditions as well. And so the way this works is the spliceosome runs down this transcript. Right? When it gets to an intron part, it loops it up, and then it cuts it off. And it gets to the next intron part, loops it up, and then cuts it off. So you have this strand of mRNA, and then you have the introns that were cut out as little loops. Okay? So after the splicing occurs, we now have our mRNA transcript, and it's almost a full-blown mRNA. It just needs a little bit more processing. Okay? And so to the front, the upstream side of the RNA, you need to add a methyl cap. And so basically it's just a methyl that's attached to the front. It keeps the RNA a little bit more molecularly sound, and it plays an important role in engaging the ribosome for translation. The poly A tail goes on the end side, the 5' side of the RNA molecule, and it's literally just a bunch of adenosines stuck to it. Right? And this tail can be of varying lengths. And so if you need a protein that the cell uses often, right, you'll have a tail on this RNA that's really long, and that allows that RNA to live for a longer amount of time, and you can get more protein translation out of that one RNA. If this is going to be a protein that's like real quick, wham, bam, thank you, ma'am, and then we're out of here, you'll have a really short tail, because then that RNA will die more quickly. And if the RNA doesn't need to be used that much, you don't want it clogging up your system. You want it to disappear as soon as possible. Okay? So the poly A tail is another form of gene expression regulation where you can control how much of a protein you're making by controlling how long the RNA will persist for. So we've moved on from our mRNA. We are now out into the cytoplasm, and we're engaging with some ribosomes to do the process of translation. Now, obviously, the mRNA needs to go inside the ribosome in order for this to happen. Right? In order to block translation from occurring, the easiest way to achieve this is simply to prevent the mRNA from entering the ribosome. And this is really easily achieved by putting something on the front of the mRNA. It makes it chonky, doesn't fit in the ribosome hole anymore. Right? And so in this beautiful model that I've made, you see our mRNA. It's ready to fit into the ribosome, but then something sticks on it and it can no longer fit in the gap. Right? It can't go on. As long as this thing is stuck on there, translation will never occur and I'll never end up with that protein. This has halted the process of gene expression. Right? So the cell changed its mind. It was like, yo, let's make this protein, and it got this far, and it was like, oh, things changed. Let's not make that protein anymore. Let's block it. Okay? And so the thing that is fitting on the front of this mRNA is another strand of RNA called a short interfering RNA, siRNA. And the siRNA is a perfect complement to a region of the mRNA. And it's tiny, hence the name, and it interferes, hence the name, great named RNA. So this short interfering RNA has a very high affinity for this region because they're perfect complements, and so they tend to hang out together. Right? They're not fully bonded the way that a DNA double strand is, right? So they just have like a really high attraction. So they kind of like stick together, almost like static attraction, right? But then they'll fall off. And so the effectiveness of this siRNA in blocking the process of translation is highly dependent on the concentration of siRNA. And so the more siRNA that the cell makes, the more effective this blocking is. And so if it wants to completely stop this process forever, it makes a lot of the siRNA. If it just wants to slow it down for a little bit, it makes a little bit of the siRNA. Right? And then you can go anywhere in between. And so there's a lot of variation in here, and there's a lot of nuances in here as well. The siRNA is coded for by the antisense strand of the gene, which makes sense, right? And so you have your gene on this side. On the other side is the antisense strand. And so like if this is my gene, the whole thing, maybe right here on the other side codes for the siRNA. And so the gene would be on the major groove side, the siRNA would be on the minor groove side, and so you can't really access it so great. You can access the full gene, and that's why you get the tiny pieces instead of the full thing. Okay? siRNA, cool stuff. And last but not least is regulation at the level of the protein. So we're just about finished with making this protein. We're just about at the end of gene expression. But the cell can still be like, nah, I don't want this protein no more, man. And we can shut it all down, okay? And it's actually really easily achieved by sticking on this tiny little molecule called ubiquitin. So my protein's coming out, it's just about finished, and the cell's like, nah, guy, we're done with you. And it sticks this ubiquitin onto the protein. Once the ubiquitin is tagged, or rather, once the protein is tagged with the ubiquitin, there are other giant protein complexes floating around the cell called proteasomes. These proteasomes are basically barrel-shaped protein complexes. And when the proteasome engages with a protein that's been tagged with ubiquitin, it takes the protein and puts it inside the barrel, and then it closes the barrel. Right? And so now this protein's like quarantined. Right? It's a quarantined protein. And as long as it's inside the barrel, it can't do any work. And so it's like it doesn't exist. Now, depending on the cell conditions and the proteasomes involved, the protein can either be saved for later when it's going to be used, right? So maybe I just don't need it right now. Things changed, but I'm going to need it next week, so I just save it till next week. Or if I'm not going to need it like ever again real soon, that protein can just be broken back down into amino acids that can be used to build another protein. So depending on what's more efficient for the cell, there are two options at this point in time. But yeah, that's it. Those are the steps eukaryotes, or those are some of the mechanisms that eukaryotes use to regulate their gene expression. Bye.